With that, I'd like to turn to our next speaker, Dr. Michael Koslowski. Michael is CMO at GammaDelta Therapeutics, and I believe you will address the utility of adoptive transfer of particularly a specific subset of human GammaDelta T cells, I believe Delta 1 cells. Michael?
Exactly, thank you. Let me tell you about the second subtype of GammaDelta T cells that Danielle and Jürgen already to the VDelta 1 subtype, and we at GammaDelta Therapeutics, we develop allogeneic cell therapies based on VDelta 1 cells. And just as a short introduction again, so when you look at GammaDelta T cells, it's actually a 500 million year old cancer surveillance system with a very unique evolution. As I said, 500 million years old. It's actually the third lymphocyte lineage besides alpha beta T cells and B cells. And, it has a very unique niche, this cell type. And this is specific to the VDelta 1 cell type. They actually generate in the thymus, they leave the thymus, and then they migrate not to the blood or the lymphatics, but they migrate to tissues. And there they reside and survive. And they have a very unique function. So they reside in tissues, and they have an active role in tissue surveillance from malignant events to recognize malignant transformed cells. But they also have a very important role in recruiting and activating other immune effectors of the adaptive immune system. And they have a very unique role in cancer surveillance. And I think this is genetic data that came up very early when you actually knock down the delta chain in mice. And mice, I have to say, only actually have functional VDelta 1 cells. In mice, you don't find VDelta 2 cells. So when you actually knock down the delta chain, then these mice develop spontaneous carcinomas. And you don't see this when you, for example, knock down alpha beta cells. So this is very specific and unique to VDelta 1 cells in mice. And when you then cross a delta knockout mouse with conventional tumor models like the TREMP model, which is a prostate cancer model, then you actually see more severe and aggressive cancers develop. So when you invite 4 speakers to talk about GammaDelta T cells and their relevance in cancer, then you have to be prepared to see this slide from all 4 of them. So you saw it from Danielle, from Jürgen, and I have a strong feeling you also will see it from the 4th presenter. I think this is definitely one of the landmark studies that came out a couple of years ago that raised a lot of interest in GammaDelta T cells and their role in human cancers, predicting GammaDelta T cells as one of the major prognostic factors for overall survival in cancer patients. So what this group actually did was a bioinformatic analysis of all immune effector cells infiltrating tumors across 18,000 patients and across all tumor types. And then following it on, when you look into individual studies, you actually confirm this. So this is work by Adrian Hayday, where he looked in triple-negative breast cancer, specifically for the presence of specific immune effectors. And here he, in this study, could confirm that the presence of VDelta 1 cells is actually highly relevant for the overall survival and outcome of these cancer patients. As Jürgen already said, there's also a lot of data coming from hematopoietic stem cell transplantation. Over the last couple of years, more than 20 years ago, Larry Lamb published these data that after allogeneic stem cell transplantation in ALL patients, the outcome and the survival of these patients was dependent on the presence of GammaDelta T cells in these patients, and they could confirm this in a follow-up study over 8 years. And then one of the latest study was actually a prospective study looking at patients receiving allogeneic stem cell transplantation, different indications. And here these results actually could be confirmed that the more gamma-delta T cells these patients actually develop from the graft, so these are allogeneic gamma-delta T cells, the better the overall survival. And surprisingly, in this study, they also showed that the incidence for graft versus host disease was actually reduced the more gamma-delta T patients develop. And since these are allogeneic gamma-delta T cells, it also shows that these cells potentially don't induce graft versus host disease. So when we talk about gamma-delta T cells, as discussed before, we're talking about two main compartments and subtypes. We're talking about blood, where you mainly find the V-delta 2 cells that Danielle and you also were speaking about. These are very easy to grow in large quantities and have been used in a lot of clinical trials over the last couple of years. When you then look for specifically the V-delta 1 cells, you only find very, very small numbers because these cells are actually tissue resident, which what you see on the right side. So in blood, you have a lot of V-delta 2 cells, hardly any V-delta 1 cells. And then when you look into tissues, you don't find a lot of V-delta 2 cells, but you find a lot of V-delta 1 cells. And we at GammaDelta Therapeutics, as I said, we focus on V-delta 1 cells for allogeneic cell therapies. And we actually have developed or are developing two platforms, blood-derived gamma-delta T cells and tissue-derived V-delta 1 gamma-delta T cells. And I would like to take you through our platforms and how we are developing and the underlying biology. So the first product I'm talking about is GDX12, which is a non-engineered blood-derived V-delta 1 T cell therapy. And when you talk about, and the process is very simple as it is. So we start with healthy donor material, leukapheresis material, which is cryopreserved. Then the material undergoes an alpha-beta T cell depletion to remove all the alloreactive alpha-beta T cells. And then we have a feeder cell-free expansion process. And after 14 days, we cryopreserve the drug product, which is called GDX12, which is always important when you do cell therapy, starting with donor material, allogeneic material. You want to have control of your donor material. So after hundreds of expansions that we actually have performed at GammaDelta Therapeutics, we have been able to identify a signature and markers that actually let us predict the outcome and the yield, the manufacturing yield from that the specific donor actually can deliver. So before we actually start expansion, we already can select those donors that give us the highest yield. And here you see that we have excellent high yield from a single run, tens of billions of cells. And we have a very high product purity. So these are 70 to 80% V-delta 1 gamma-delta T cells. And you see that we hardly have any alpha-beta T cells in this product. We have a very nice functional phenotype after expansion. So we see high expression of CD27, high expression of NCRs, natural killer receptors, and very low expression of typical checkpoint molecules on these cells. Although I have to say that the role of these checkpoint molecules, specifically in V-delta 1 cells, is still not fully understood, and they seem to function differently than they do in alpha-beta T cells. When you look at the fitness of the drug product, we have very high recovery post-cryopreservation. For many cell therapies, it's actually difficult to develop a cryopreservable product. And what is also interesting for clinical translation is that these cells, once you thaw them and give them in culture again, they actually start proliferating again. So that's all speaks for a good drug product fitness. We have a very safe cytokine profile. So our cells, irrespective on how you stimulate them, if you do TCR stimulation, cytokine stimulation with target cells, they have a Th1 focused cytokine profile. We don't see any cytokines that would be associated with cytokine release syndrome. And specifically, we never have seen IL-17. So there is always data and publications around gamma-delta T cells, B-delta-1 cells that express IL-17. In our studies, we have never found a human B-delta-1 cell that actually expresses IL-17. There are IL-17 expressing B-delta-1 cells in mice, but so far in humans, we have never observed any of them. And this is then what we see regarding efficacy. These cells very potent and selectively kill cancer cells. In this is an AML cancer cell line. We see the same when we look at primary patient blasts. And what we always see is when we look at healthy allogeneic PBMCs, we don't see any activity. So these cells show an intrinsic tumor-selective cytotoxic potential. And just to the clinical translation, in vivo, we also see good tumor control. This is a NALM-6 ALL xenograft model where you see the control group growing out. And after a single dose of GDX12, in these, I have to say, suboptimal models because these are NSG models where you need to supplement cytokine externally. You see good tumor control. And GDX12 is now our first product that we have brought into the clinic. So we have initiated a clinical study in Q3 2021, and it's currently tested in patients with MRD-positive AML. We also have received orphan drug designation for the treatment of AML, and we are currently in dose escalation. We are treating between 40 million and 4 billion dose levels, cells, 4 dose levels, and then we are planning to run a dose expansion cohort at the maximum dose level. So this is the unmodified GDX12 blood platform, which for us is also a plug-and-play platform to actually develop our gene-engineered cell products. And as a proof of concept for our blood-derived cell type, we have selected CD19 as a target because it's well precedented. The process is very similar to GDX12 with the transduction step in between. So again, we start with leukapheresis material, healthy donor selected. We do a alpha-beta T cell depletion, transduction on day 4, and then again, we have a feeder cell-free expansion process and have a cryopreserved drug product. Here you can see that we have very nice CD19 CAR expression in these cells. We have excellent viability of untransduced and also the transduced cells after cryopreservation. And then in vivo, and this is in vitro, we see when you look at the untransduced cells or GDX12 against NALM-6, you see dose-dependent with higher effector target to ratios increasing cytotoxicity. So there is some baseline cytotoxicity of GDX12, obviously on ALL cells also. But when we then transduce these cells with a CD19 CAR, we see a significant increase in cytotoxicity, specifically at lower effector to target ratios. What we then analyzed is the cell expansion upon target recognition. So when we actually run killing assays over multiple weeks, repeat antigen stimulation assays. This is an assay over 2 weeks. We have extended this up to 3 weeks, where we actually replace the tumor cells every 3 days. So after every killing round, after 3 days, the cells are exposed to a fresh batch of tumor cells. And then you can see that actually the cells are expanding upon tumor recognition and killing. And what we also see that we keep a very functional phenotype. So we see some upregulation of PD-1, CTLA-4 or TIGIT, but we don't see any loss in cytotoxicity. So there is no decrease in cytotoxic activity comparing the first killing round to a killing round after 3 weeks of repeat antigen stimulation. And what we're also doing with our gene-engineered products, we armor these products with cytokines. So we have a cytokine armoring strategy developed. so that these cells actually, when they are armored with cytokines, show better and enhanced survival in vitro and in vivo without any exogenous cytokine stimulation. When we test GDX14 in vivo using an ARM6 tumor model again, you see the control group where the cells are growing out. And then when we treat these mice with two different constructs this time, I call it binder 1 and binder 2, you see that in this model, we treat with 2 million CAR T cells, and they are probably transduced to 30%. So we have roughly 600,000 CAR T cells here. And we are actually treating pretty late. So after tumor injection, we are injecting the CAR T cells only on day 7, after the tumor is well established. And then you see that with our selected binder that we selected to take forward, we actually have complete tumor control. And when we then run a re-challenge model in vivo, this is also nice data that actually show that these cells also persist in vivo. We run a first model, again, treated after day 7. You see outgrowth of the control group. You see complete tumor regression in the treated mice. And then after day 19, we treat these mice again. We dose them again with NARM6. And then you see again in the control group the outgrowth of the NARM6 model, but in the treated group that was only treated on day 7, you see that there is no tumor growing out again. So we think that we have a very potent allogeneic CD19 product that we are planning to develop. Looking at genetically engineering these cells, the question is, can we do more? So developing CAR T cells for hematologic malignancies, but also for solid tumors specifically, is always dependent on the presence of, on the availability of very specific targets. And then the problem is, because these targets are hardly available in solid tumors, that you always have to account for on-target off-tumor cytotoxicity and activity of CAR T cells. So can we actually use the unique biology of Vδ1 cells to develop tumor-selective CAR cells? And this just highlights what already was discussed in the previous talk. So gamma-delta T cells recognize an abundance of natural killer receptor ligands that are expressed on tumor cells, but they are absent from healthy cells. And the Vδ1 cells specifically, and this is, I think, very different to Vδ2 cells, the TCR itself is actually not important or not relevant for the cytotoxicity. So engagement of the TCR with a target is important to provide survival signals in tissues, but it is not relevant for the cytotoxicity. So when you knock out the TCR or when you block the TCR, you don't see any loss of cytotoxicity. So for the Vδ1 cells, the cytotoxicity primarily seems to be driven through recognition of the ligands for the NCRs that are selectively expressed on tumor cells. Another question is, can we actually modulate and modify the function, the signaling domains of a CAR that we're inducing into VD1s to maintain this selectivity on tumor cells and spare healthy cells? And what we observed first when we tested different CAR constructs is the following. So when you look here, the CAR19 version 1 is a conventional second generation CD19 CAR with a 4-1BB CD3 zeta signaling domain as it was developed for alpha beta T cells. And then we have a second CAR where we have optimized the signaling for Vδ1 T cells. And you see both in vitro, both CAR constructs show the same increase in cytotoxicity compared to a mock transfected Vδ1 cell on NARM6 tumor cells again. But then when you look at the cytokine production of these cells, what the interesting thing is, when you look at the second generation CAR construct, you see high cytokine release, interferon gamma, TNF alpha, IL-2, MIP-1 beta. But when we modulate to V delta 1 biology, we actually can abrogate cytokine release from gamma delta T cells, but maintain the same cytotoxicity. So, and we think that this opens up a lot of opportunities to develop CAR cells with better safety and that are more tailored to V delta 1 biology. And this is just one example, another example that I want to show you. What we have done here is, and the question is really, can we tackle on-target off-tumor activity with these V delta 1 CAR T cells that you normally see when you would use an alpha beta T cell, where all the cytotoxicity is just driven by engagement of the CAR with a tumor target that also might be present on healthy cells. So what we have developed is a tri-culture cytotoxicity assay where we incubate and culture healthy B cells that express CD19 with NALM6 cells that express CD19 with our product, the gamma delta T cells. So when we do this again with our mock transduced GDX12 cells, the non-transduced gamma delta T cells, what you see is that these cells bear at all effector target to target ratios the normal B cells. And they only kill the NALM6 cells. When we now introduce a second-gen CD19 CAR construct for 1BB CD3 zeta, what you see is that we have a very nice increase in cytotoxicity compared to the mock transduced cells. And we also see an increase of cytotoxicity on B cells. But what is really interesting to see is that the cytotoxicity of B cells is not equal to the cytotoxicity observed on the NALM6 cells. So there is definitely already a window of opportunity that you might spare normal or have less toxicity on normal cells. And it probably also shows that the engagement of the NCR is still relevant to maintain full cytotoxicity. So what happens when we now fully optimize a CAR construct to V delta 1 biology? This is what we call the next gen CAR constructs. We still see the very high cytotoxicity on NALM6 cells, but we then nearly completely abrogate cytotoxicity on normal B cells. So what this shows is that we can actually use gamma delta T cells, specifically the V delta 1 cells, to potentially develop CARs that are truly tumor selective, and that you could broaden the target space with these cells, going for targets that are not highly tumor selective, but so-called dirty targets that might be expressed on toxicity relevant normal tissues. So this is something we are obviously very excited about, and we are testing this now across our different platforms to further optimize. And that will also help us to optimize signaling domains, specifically optimized for V delta 1 cells to promote persistence and proliferation of these cells. And the last slide, just very shortly, to the skin-derived V delta 1 cells. So what I showed you before is all based on our blood platform. And as I said earlier, most of the V delta 1 cells are actually tissue resident. And what we are currently investigating is if the tissue resident V delta 1 cells actually also can be utilized to develop cell therapies, because we think that tissue resident cells potentially might be better suited for the treatment of solid tumors, because they infiltrate tissues, they are adapted to survive in tissues, they receive survival signals in tissues via their TCR. So this is something we're currently investigating. And this is how it looks. We take fresh skin, we take punch biopsies from the skin, we load them on grids. These are specific grids manufactured to maintain organotypic cultures in vitro, and then we have a culture process to expand these cells. And you might imagine that this is quite a cumbersome process, and it's actually not feasible to start every manufacturing route just from a piece of skin, taking biopsies and then running this process. So what we have decided to develop at GammaDelta Therapeutics is something that we call DermaPack. So we actually generate qualified starting material from skin. This is a 3-week process. And then we actually freeze it down. And we call it DermaPack in analogy to the Leukopack. So we have here actually a GMP-enabled cell bank of skin immune effector cells that is allogeneic, where we have donor selection, in-process control, and cryopreserved, that we can then take as starting material to expand and transduce our cells. And although this is currently tailored to expand V-delta-1 cells after this initial step, we already have proof of concept data that this also can be skewed towards other immune effector cells that you would want to isolate from tissues or skin. So this is a new product per se that we are using to develop our skin-derived GammaDelta T cell platform. And just some initial data that also show you why we believe that this is a platform with potential. First of all, freezing down and then expanding and transducing these cells again results in a product with high viability and recovery. But what we're really excited about is when we've run first in vitro experiments using a specific tumor target that I cannot disclose here, but this is a CAR generated against a tumor target that is expressed at different levels in different cell lines. So when we use an alpha-beta second-generation CAR against these tumor targets, we see good killing, effective killing. But then you see when we just use the mock-transduced V-delta-1 cells from skin, they are not expressing any CAR. They are non-transduced. They are already equally potent to an alpha-beta CAR on these cell lines. And then when we introduce a second-generation CAR, again, 4-1BB CD3 zeta, you see an increase in cytotoxicity, roughly 20%. Which just highlights that these cells also have a very unique biology, which is very different from the blood V-delta-1 cells, where with a second-gen CAR, we see a massive increase in cytotoxicity. But in skin-derived cells, we don't see this massive increase. There is some benefit, but we think this opens up for us opportunities really to understand the difference between skin and blood-derived gamma-delta T cells. And we are currently performing a lot of comparative studies to understand what the true difference between these cells is and how we can further optimize the CAR constructs to even further increase the cytotoxicity of the skin-derived cells. And that's actually my last slide. So I hope I could show you how we are trying to develop truly differentiated platforms for adoptive cell therapy. And thanks for your attention. And I'm happy to answer any questions in the panel session.